A warm good morning to all of you. I feel honored to welcome you all to the international webinar on entomology, organized by Shadpada Entomology Research Lab, Department of Zoology, Christ College, Iringalakota. Let me introduce myself. I'm Dr. Bijoy, Assistant Professor, Department of Zoology, and Principal Investigator, Shadpada Entomology Research Lab, SCRL. Though we are living in the midst of a pandemic with uncertainty, we still find opportunity to share global scientific ideas through virtual platform in the form of webinars. This event is the second of such efforts from our department. Once again, I welcome you all to this visual gathering. First, let me invite Dr. C.O. Joshi, Vice Principal and Head of the Department of Zoology, to deliver the welcome address.
So, one good morning to one and all. Welcome to the members of Christ College Autonomous Iringalakota. I take this opportunity to welcome all the distinguished guests and participants of this international webinar. At the onset, I would like to tell you something about the activities going on in the department. Department of Zoology took its birth along with the birth of Christ College back in 1956. Within a short span of 5 years, the department was raised to the status of postgraduate department, and in 1973 achieved position of research department in the department of under the University of Calicut. Right now, we have 12 research scholars in various sections, along with another 24 in physiology lab, immunology and toxicology lab, entomology lab, then biodiversity lab, neurobiology lab. A total of 36 research scholars are now with under the guidance of 12 research guides. The strength of the department started by the activities of the very first principal of Christ College, Reverend Father Padma Bhushan Gabriel Chiramel CMI. He was the first HOD of Department of Zoology. And we had a prominent professors in the department. To mention a few, Dr. C.K. Nair, Dr. E.G. Govindangutti, Dr. V.V. Sudhakaran, and the like. Under the leadership of those eminent personalities, professors, department grew in size and visibility. And right now, we are in this position of 12 research guides and 36 research students. The department has already produced 25 PhDs and 2 are waiting for their open defense. With this brief introduction to the Department of Zoology, Christ College, let me get into my duty. Let me welcome the most respected principal of Christ College, who will be inaugurating this session immediately, Reverend Father Dr. Jolie Andrews CMI, a professor in the Department of Physics, who has been showing keen interest in research and who has been publishing research papers in various international journals continuously with very good citations and text. He is with us with all respect. In the name of the department and in the name of the organizing committee of this international webinar, I welcome you, Father. Welcome, Reverend Father Principal.
Thank you. Thank you so much.
Now I will. Thank you, Father. Now I welcome our resource person, Dr. Rebijit. Dr. Rebijit hails from Balusheri, a suburb area of Kozhikode. And he has his PhD work in India only, and then reached New Zealand a few years back, and he is working as a senior scientist with the Ministry of Primary Industries, Auckland, New Zealand. The topic selected by him or suggested by him for the international webinar is the perspective of molecular approaches in entomology. This is really a new area of research, or an area which is demanding a lot of effort on the part of people or scientists and research scholars in the field of entomology. This is not mere taxonomy or mere work with regard to the classical taxonomy, but something very important. And this topic has already proved that this is one of the best he can provide us, because we have more than 400 registrations till now. It's a huge number of registrations. We are very happy, primarily because the topic of Dr. Rebijit is rather very, very significant. In the name of Christ College, in the name of Department of Zoology, Christ College, in the name of the organizing committee and all the participants, Dr. Rebijit, I welcome you to this international webinar. Dr. Rebijit.
Thank you.
Okay, sir. Okay, sir. Now, I have great pleasure now inviting all the executive committee members, right from the patron of the international webinar, our respected manager, vice principals, deans, and the IQAC coordinator, Dr. Robinson Paminchery. He has been with us for this seminar, and every technical support has been extended to us by Dr. Robinson. I welcome Dr. Robinson. Okay, now I welcome the faculty members of the department and all the research scholars of the department. Finally, every seminar is successful only with the participation of the most interested participants from everywhere in the world. And I welcome the participants in the name of this organizing committee, and I also welcome Dr. Bijoy C., Assistant Professor in the Department of Zoology, Christ College, who is the organizing secretary of the seminar. Welcome, Dr. Bijoy. Now, with this brief introduction and with this brief welcome, I invite our respected principal, Reverend Father Jolie Andrews CMI, for his inaugural address. Father, please. Thank you.
Okay. Am I audible? And can you all see me? Okay. Thank you so much.
Yes, Father.
Thank you. Thank you so much. Most respected Dr. C.O. Joshi, the Vice Principal and the HOD of the Department of Zoology, is the convener of this international webinar, Perspective of Molecular Approaches in Entomology. The resource person of the day, Dr. K.B. Rebijit, the senior scientist from Auckland, New Zealand. the organizing secretary, Dr. Bijoy, most eminent faculty members of the Department of Zoology, the faculty members from different parts of the world, research students, and my dear friends. I'm very happy to inaugurate this international webinar that has been organized by the Department of Zoology. I appreciate and congratulate the efforts that has been put forward by the Department of Zoology in helping the researchers and the scientists from the different parts of the world to come together to share their ideas, which will become definitely a motivation for the researchers, for the students, and for the faculty members. I'm happy to note that today, many of you are participating in this international webinar on the topic, the perspective of the molecular approaches in energy. To Abhijit KB, Dr. Abhijit KB, who himself is a senior scientist from Auckland, New Zealand. A word also about the Christ College, as has been mentioned by our Vice Principal, Dr. C.O. Joshi. The college was started in 1956 by the legendary Father Padma Bhushan, Father Gabriel, who himself was from the Department of Zoology. I am happy to note that the Department of Zoology is leading the list of the PhD holders that has been awarded by the Christ College. The number which has been here mentioned by the Vice Principal and the HOD of the Zoology, Dr. C.O. Joshi. As far as the Christ College has been concerned, we are perhaps the college which is having enrollment of the largest number of the students. Last year, our enrollment ratio, the student enrollment was around 3,900. One of the most sought after campus due to its biodiversity, due to its lush green, environmental friendly, and student friendly, and the research promoting atmosphere that we promote here in the Christ College. We are definitely having a rush of the students to this campus, since this campus has become an epicenter of an academic pursuit and learning as far as the quality has been concerned. So once again, I appreciate very much the Department of Zoology for coming forward here. I'm happy to note that I extend my appreciation to the organizing secretary, Dr. Bijoy C., and all the other faculty members, eminent faculty members, who have been shown their efficiency as far as the research has been concerned by getting Young Scientist Award, by getting other DST projects and everything. And I would also like to say that we have been having the largest amount that has been from the, as far as the college has been concerned, that has been contributed by the Department of Zoology. In those way, I very much appreciate all the faculty members of the Department of Zoology in a very special manner. I wish you all the best. Have a wonderful day. May God bless you. And I officially inaugurate this international webinar. Thank you.
Thank you, Father. Thank you, Father.
Thank you, Father. Before we begin, I have a few instructions to the participants. Dear participants, always keep your microphone mute and video off. Please text your queries in the chat box. Our team will be passing the question to the speaker at the end of the session. And please don't post any kind of greetings and other messages there. A feedback form will be provided at the end of the session, both in Gmeet and also in YouTube live stream. You can fill it correctly to get your participation certificate. If you have any technical issues in the Gmeet live, you can join the YouTube live stream. The link will be there in the chat box and even in your mail, registered mail. So please cooperate. Try to keep your microphone mute and video off. So now I feel immense pleasure to introduce our resource person, Dr. Rebijit. Rebijit completed his master's in biotechnology from Bharathiar University, Coimbatore. He started his career in the ICAR, Indian Institute of Horticultural Research, as a senior research fellow and completed his PhD in biotechnology from Kuvempu University, Shimoga in 2015. He won the Alfred Russel Wallace Award 2016 for his doctoral work from Royal Entomological Society, London. After his doctoral degree, he joined as a postdoctoral researcher at the Department of Physiology, Development and Neuroscience at the University of Cambridge. Later in 2017, Rebijit joined as a senior scientist at the Ministry for Primary Industries, Auckland, New Zealand, and continuing his work in the field of molecular entomology at MPI. He is passionate about applied entomology and has been leading various research projects on molecular entomology. Last year, Dr. Rebijit won the prestigious Japan Researcher Award 2019 from Japan International Research Center for Agricultural Science for his outstanding achievements of research in agriculture and related industries in developing regions. Rebijit has more than 36 international and 9 national publications, and he is an author of several book chapters. He is also a serving editor and reviewer for various research journals and participating in various entomological society activities across the globe. Apart from many other research projects, Rebijit is currently leading an interesting project entitled Life After Death, using RNA as a pinpoint time for the death of insects. So participants, please join me in welcoming Dr. Rebijit. And Rebijit, the session is all yours.
Right. Thank you, Vijay, for a nice introduction. Yeah, it's good to see so many people. Yeah, so I am just opening my screen. So can you see my screen?
Yes, sir. Yes, sir.
Okay, right. Thank you. Yeah, so as Bijoy and the department head has mentioned already about the topic, it's perspective of molecular approaches in entomology. So yeah, as Bijoy mentioned, that I'm currently working as senior scientist in molecular entomology at Plant Health and Environment Laboratory, which is based in Auckland for Ministry for Primary Industries. So yeah, as most of you are from zoology background, you might be knowing entomology, but let me start with entomology. So entomology is nothing but study of insects and their relationship with humans, plants, and environment, and indeed with other organisms as well. So as all of you know, like, why is it important? Because insects are considered to be the most abundant animals which is on Earth. Okay, almost 1 million different types of insects has already been identified so far. And another important feature of entomology is insects are like they can survive in any situation, like in extreme cold and hot condition. And apart from that, most of these insects, which they are severely harmful as direct pests, apart from that, they have the capability of vectoring some of the plant viruses. And apart from all of these things, like, we have to understand that among the insects identified so far, some of the insects are really beneficial as well. Say, for example, in case of honeybee. Okay, so yeah, this is about general introduction about entomology. So you might be knowing like different the way of like it can be classified in different orders, like something like Lepidoptera, Diptera, Hymenoptera, Coleoptera, and so many other things. Right. So I'm divided my today's talk into two aspects. One is on identification and classification, and the second part of my talk will be on the management. Because as I mentioned in the previous slide, that most of these insects are harmful pests. So we need to know how to manage them. Okay, then only we can survive in this world, because as we know, like most of these insects, as direct, it can damage agricultural crops. Okay, so we need to have different ways to manage these insect pests. So coming to the first part of my talk, like where I'm going to talk on identification and classification. So as all of you know, like compared to any other organism, like insects also been classified as into genus and species, which is basically from Linnaean system. So apart from that, there are three main important classification system as of now, which is available. So the first one is again Linnaean system, which has been put forth by Carl Linnaeus. Okay, so this is the book which he wrote on the how the classification system should be. The entire book is known as Systema Naturae, and this is the most accepted system which is in place at the moment. So say for example, in case of humans, so we know that the scientific name is Homo sapiens. So we have a genus name followed by a species name. Okay. So apart from that, we have two other systems which is in place. The first one is cladistics, where again we'll try to organize or classify organism based on its traits. Say for example, apomorphy, plesiomorphy, autapomorphy, synapomorphy, homoplasy, all sorts of things. So say for example, apomorphy is nothing but a novel evolutionary trait that is unique to a particular species, and it's all of its descendants. Say for example, in case of birds, okay, they have like feathers. Okay, so feathers of birds are unique character for all of that class, which is known as aves. So yeah, similarly, we can again define all of these different systems. So it's existing, but which has not been well utilized by many, and that is known as cladistics. Okay, and the third and the slowly growing branch of classification system is the phylocode. So it's in other term we can call it as phylogenetic nomenclature. So in order to work in this, there is a big branch now which has been known as International Code of Phylogenetic Nomenclature, which has been formed by a big group. Okay, so yeah, these are the major three different classification system as of now which is available. And apart from that, as I already told you, like the Linnaean system is the most accepted and most widely used. But before going into the classification and identification, all sorts of things, we need to understand why we need a really system in place. Okay, so with the classical like taxonomy, or what we can call like the present identification system in place, which took almost 200 years to count 1.7 million species. Okay, and apart from that, though we took this many years, we have almost counted less than 10% of the estimated species on Earth. So apart from that, we have less funding for classification and identification studies, and dwindling interesting taxonomy is another problem what we are facing. And apart from that, increased international trade causes a risk of introduction of invasive species to many countries. Okay, and accelerated extinction rate of various species is another reason where we need a better system in place. And of course, as all of you know, the most important, which is very important from all of our perspective, is to monitor the biodiversity, and also we need to conserve them very properly. So if we are taking too many years to identify and classify, so many species might extinct before we count them into a real name. Okay, so apart from that, as I told, like we have some system which is already in place. Say for example, classical taxonomy. So we need to understand what are the shortfalls in morphological identification in order to bring another system which can cope up or which can replace all these shortfalls. So as I pointed out here in the slide, so you can see like phenotypic plasticity is the first one, which might be really difficult to work with morphological identification. And of course, genetic variability is not possible to count with morphological classification. And again, something we can corroborate with phenotypic plasticity. Another one is morphologically cryptic taxa, which is really difficult to like count by using morphological identification, which I'm going to talk on the more cryptic taxa in my coming slides. And many of the keys, identification keys, which are effective for particular life stages. Okay. Apart from that, sympatric and allopatric variation might be really difficult to distinguish by using morphological identification. Of course, when you need to do a morphological ID, you need a specialist, and we need a holotype, or as a whole, the whole specimen is required. So that's another shortfall. And yeah, another one which might be really difficult with classical taxonomy is identification of biotypes. Say, for example, in case of whiteflies, there are several biotypes which is available in case of Bemisia tabaci, so which might be really difficult to count based on morphological characters. So there comes a new attributes or new requirement. Okay, so as I mentioned that our planet Earth is home to 10 to 100 million species, and many of them has not been counted yet. Okay, and our human minds or human brain has an inability that it can call or recognize and recall perhaps 1,000 species. So which require a new technology where it can count faster, but at a better pace, so that we might not lose many of the species which is existing currently on Earth. Okay, so there comes the importance of DNA barcoding. So then we need to know what is DNA barcoding. So DNA barcoding is nothing but identification of species based on a particular DNA sequence. Okay, and it can be used to recognize any of the a particular domain of life, either in eukaryotes, as whether it can be used some of the DNA sequences can be used to identify in plants as well as in case of animals. So say, for example, then we need to know what kind of DNA sequence we can use. So generally that is known as markers. So these markers can be of several types. Okay, say for example, in case of animals, so the DNA barcoding region, which has been well defined as against in plants. So in case of animals, and because I'm doing my work on insects. So for most of my work, we generally use mitochondrial cytochrome oxidase 1 gene, which is from the mitochondrial organelle. Okay, so I guess you might be knowing we have, in case of animals, we have nuclear DNA, which is from the nucleus, and whereas at the same time we have an extra chromosomal material, extra nuclear material, which is residing in the mitochondria. Okay, so that kind of DNA fragment, which is known as mitochondrial DNA. And within mitochondrial DNA, there are several markers which is available for, it can be used as a DNA barcoding fragment. So something like CO1, site B, 12S, 16S, and there are so many markers which can be used as a barcoding region. But the most efficient and the most widely accepted one in case of animals are the COX1 or CO1. Okay, so by using this particular DNA sequence, we can identify a given species without much of a hassle. And similar to that, in plants, there are combination of various markers which generally they tend to use for the identification of insects. And similarly, in cases of bacteria, fungi, and protists, researchers can use various different markers, as I indicated in the slide. So as I indicated, apart from the nuclear DNA, every animal cells, they have organelle called mitochondria. So within mitochondria, we know that there is an mitochondrial genome is existing. And within this mitochondrial genome, there are, as I pointed, there are several marker genes which is available. But for the barcoding purpose, or from the molecular identification perspective, the most trusted and the most widely accepted one is the CO1, or mitochondrial cytochrome oxidase 1 gene. Okay. So now we need to know, can this technique overcome all the shortfalls which I previously mentioned in case of morphological identification? So we need to look at what are the advantages if you are using this DNA barcoding technique. So the first one is they are uniform, because since we are using a particular gene, particular DNA fragment, it can be uniform across the kingdom. at least in case of animals. And they are, another very important thing is they are life stage independent. Okay, so we need not require a fully grown adult specimen for the identification purpose. Even if we are getting a small tissue from the leg, it's possible to identify the specimen very easily. Apart from that, we can also use it for conservation biology and biodiversity studies very effectively, because in case of biodiversity, now most of the institutes or most of the researchers are using this technology very much, as the metabarcoding is very much available because of the advancement in the high throughput sequencing technology. And also this technology can be used to determine the food webs. Apart from that, we can, by using DNA barcoding, we can identify biotypes, and also it's possible to understand the phylogenetic perspective of various species which is under study. Okay, and also here in New Zealand, we use DNA barcoding for the purpose of biosecurity. So I will explain a bit more as on when we move forward. Okay, and of course, biosecurity and quarantine. Okay, and another important thing, like advantage is, which is this technology is very cost effective compared to the classical taxonomic method. Okay, so now we need to know, like, how is this working as a whole. Like, say for example, in case of insects, so we will be having specimens with us, and from there we'll collect a tissue, small part of a tissue. So otherwise, if it is too small, we can use the whole specimen as such. But generally, we tend to do like good laboratory practices where we use a small portion of the tissue, and the rest of the specimen will go as a voucher specimen, so that further taxonomists can look into it and they can identify them properly as well, if it is not possible with the molecular technique. Okay, so otherwise also we can keep the specimen as a voucher specimen, so which is very important nowadays. So what we do is, from the specimen we'll extract some, like we'll take some tissue sample, and from there we'll extract the DNA, and then we'll go for PCR amplification, and from there the amplified product can be sequenced by using Sanger's sequencing method. So apart from that, once we receive the sequencing, so what we can do, we can either do a blast in BOLD database, which is the known barcode of life database, so where millions of sequences have already been submitted by various researchers across the globe. This was initiated by Canadian research groups from Ontario University. So where you can, apart from the sequence submission, or apart from the sequence row sequences and the rest of the details, you can also submit the collection details, photographs, and other data into this web domain. Okay, so that will be useful for the researchers who are doing research across the globe. Okay, and as I mentioned, like, say for example, an unknown insect, when we are doing, we can do a simple blast and we'll come to know like which species it belongs to. So that's how it works. Okay, apart from this BOLD database, we have something in some other website, web domain or public domain, where we can do blast option is NCBI, National Center for Biotechnology Information. There also we can do a blast hit, and we'll come to know like which species we are working with. So this is the general introduction of that topic. And now I'll just show you some of the application which I did while I was in India when I was doing my PhD. So where I did my work mostly on thrips and aphids. So with regards to thrips, we generated almost 1,000 sequences, which is representing around 151 species. And of course, like when we looked into the nucleotide frequency, we came to know like it has been biased towards to adenine thymine combination with a total of almost approximately 70% nucleotides. Okay, and apart from that, it like most of the species, like the intraspecific distances were ranging from 0 to 10%. So that was quite amazing. And from the analyzed 151 species, all of them were clearly able to discriminate based on the COR1 sequences. So that indicates that COR1 is a very stable marker in case of thrips to distinguish them as into a species level. But however, genus thrips and Franklinella had shown the paraphyletic nature when we created the phylogenetic tree. And for all of our work, we used the same primer, which I was mentioned in my previous slide, that CO1, so which has been which can be amplified by using the primer LCO 1490 and HCO 2198, which has been proposed by Hebert et al. In 2003, and which has an amplicon size of 700 base pair. So this is the phylogenetic tree for the 151 species which we have studied. So here you can see Terebranchia is a big group in Thrips, Thysanoptera, and Tubulifera is another group. So both of these group has clearly able to distinguish based on the barcodes which has been generated. Apart from that, all of the species which were clearly, reliably were able to identify based on the DNA barcodes which has been generated from our study. However, we ended up like some interesting result from our study when we compared the sequences of Indian thrips with the rest of the world. And one of such interesting study was like the thrips palmi from Indian population was showing an unique clade compared to the rest of the world. Okay, and even in there, couple of sequences were there from India, but it was from the border with Pakistan. So we don't know whether it's really from India or from Pakistan. So, but anyway, we concluded that in our study, we concluded that the Indian population was showing some clear-cut differences, and the Indian population of thrips palmi, because we really don't know how far it's been studied abroad, but in Indian population, in Indian thrips palmi, which has an enormous potential in transmitting a vector, a virus, which is known as watermelon bud necrosis virus in India. So this was telling some story behind it. So yet we couldn't able to do that kind of virological studies, as we were unable to import any of the specimen from rest of the world to India to do such kind of studies. But however, we presume that it could be showing some kind of significance. That's why the Indian population was standing out with the rest of the world when we compared the sequences. So similarly, it was not only limited to thrips palmi, but apart from that, we could able to clearly distinguish five other species apart from thrips palmi: Thrips tabaci, Sitothrips dorsalis, Thrips hawaiiensis, Sitothrips persei, and Franklinella occidentalis. So what we understood was, when we done more research onto it, we understood that the intraspecific distance were much higher compared to the intraspecific distances. So what it means, like, in Hebert, when Hebert has proposed the CO1 as the barcoding primer or the barcoding region, he also postulated another theory which is known as 10x Hebert's rule. So which he popularly placed in case of Lepidoptera, but we adopted that rule in case of Thysanoptera and proposed that all of these 6 species can be classified as a cryptic group in case of thrips. That means, again, which has no which is not possible to identify by using morphological technique, but however, they were clearly showing that there are different, indeed they had different clades or different groups when we are looking at its genetic structure. Okay, so yeah, the same has been published in Florida Entomologist, which has been published in USA. So yeah, that's what the entire study which we done on thrips. But coming to the studies which we conducted on aphids, where again we could able to like distinguish several species. But before going into that, when we are doing this work, it was mandatory. Like, some of the farmers came to us and told that some of the aphid species were co-occurring on host species. So is there any way to identify them properly at a very early stage? So we were finding like it was really difficult if it's in very early stage, while like very early instars or nymphal stages, it might be really difficult to identify them to species. And thus we came up with another interesting technique, which is known as species-specific primer. Okay, so in order to design the species-specific primer, what we did was we done the DNA barcoding. Say, for example, here we proven that like we took the examples of Apis gossypii and Mysis persicae, which can co-occur on many crops. So what we did was we aligned the sequences, and we understood where the differences are relying on this in this species. And based on the differences which is existing in certain places, we designed primers unique to both the species. Okay. Again, the entire study was conducted by using Herbert's primer, which is of 700 base pair. So thus, we designed several primers for within the sequences, as I shown in the previous slide, and we differed the product size. Okay, so say for example, in case of Mysis persicae, I have designed a primer in 43 to 70 as a forward primer, and I designed a reverse primer in 450th position. But at the same time, I retained the same forward primer for Ephis gossypi, but whereas I changed the reverse primer to a different site, thus the product size varied. Okay, so say for example, if a farmer is bringing a sample to our lab, we need not have to go for sequencing. We just need to do the genomic DNA isolation, which is pretty possible within 20 to 30 minutes, and then we need to complete the PCR, which is pretty possible with 1.5 hour time. Okay, and then we need to run a gel, which is pretty possible within 10 minutes. So we can provide the information which species it is just within a span of 2 hours. That is what the importance of this work. Okay, so based on that, just by doing a PCR and just agarose gel electrophoresis, we'll come to know, like based on the product size, we will come to know which species it is. Okay, and we need not wanted a fully grown adult specimen to identify the species. And such kind of, we developed several of such species specific marker, like as on when the requirements was there. And this particular paper has been again published in Florida Entomologist, published from USA. And apart from that, we did quite a lot of DNA barcoding studies in case of aphids. So we could able to complete DNA barcoding for almost 40 species, which has been collected across India and from a series of crop plants. And another thing is, like, similarly what I mentioned in case of thrips, we could able to identify some of the cryptic species here as well. So you can see, like, the intraspecific distance, the interspecific distance was 10 times higher than that of the intraspecific distance. Okay, so here the 3 species which we were identified was Hyperomyces caudilloneus, Brevicoran brassicae, and Brachycardus helichryssi. Okay, so all of these were within that 10x Herbert's rule, and that's why we proposed it as cryptic species. And again, the same paper, the barcoding paper and elucidation of cryptic species, has been published in Bulletin of Entomological Research, which is published from Cambridge University Press. And after that, we thought that, okay, we need to do something which is more interesting. So then what we did was, as on when we collected several species across India on a number of host plants, and also they were all falling in different tribes, subtribes, family, subfamily, and all of them were comprised into a superfamily, which is known as Aphidoideae. Okay, so then what we did was, like, apart from the COV1 gene alone, we thought of studying the evolutionary history of aphids. And then what we did was, apart from the COV1 primer, COV1 marker gene, we also went into another 2 markers, which was, again, one was from mitochondrial genome, which is known as COD2 region, and another one was from the nuclear gene, which is known as elongation factor. So why we use a combination of molecular markers is, generally it is known that when we are using the mitochondrial genes, it tend to evolve very fastly. Okay, so when we are doing evolutionary studies, it is not appropriate if we are doing only if we are calling the phylogeny based only on such kind of fastly evolving gene. So if we can combine another 1 or 2 nuclear gene, which might not tend to evolve such quickly. So if we can combine both these things, such kind of phylogenetic inferences will give more information compared to the phylogeny which has only restricted within mitochondrial genome. So here again, our studies has contributed to understanding for a better understanding of the higher level relationship of Aphidoideae. Okay, and we have been, like, in this, by using this study, we have shown various sister group relationship within the group. Like say for example, in case of Ephidinae and Macrosiphinae, and apart from that, Fordinae and Pampyginae. So there are several sister groups were revealed, and also we also done the molecular dating of divergent studies, where it has showed that most of the diversification has occurred in the Cretaceous period, late Cretaceous to late Oligocene period. Okay, so these were some of the conclusion from that particular studies, and the same was published in 2017. So these are those things which I was talking on my previous experiments or previous work, and whereas now I am working like on a quicker, like we need to identify things at a quicker pace. Okay, so for that, nowadays we used to develop some of the assays which is based on real-time PCR methods. So here what we do is like we design primers and probes, okay, which can give real-time answers for the species in question. So what we do is like, say for example, in this particular study, we were developing an assay which is specific for Bacterocerca cucurbitae. So what we do is initially we do sequencing, barcoding or whichever the gene of your interest. Generally, we tend to do barcoding because there are several sequences which is already been deposited in NCBI and BOLD database. So we do barcoding, and then based on that sequence, we develop forward reverse primers, and apart from that, we'll use a probe. Okay, so as on when the primer extends, the reporter will give us signals, and that can be captured by this real-time machine very efficiently. And then we'll do a series of experiments based on all of these parameters, something like probe concentration. We need to know like what concentration, like how much amount of probe is required to give accurate signal. Apart from that, BSA concentration, primer concentration, MgCl2 concentration, probe mix, annealing temperature, sensitivity testing, all these parameters will be extensively studied, because such developed assays, real-time or PCR based assays in our lab used to go for accreditation. Okay, so at that time it is very important, because all of such assays are maintained at a higher level of accreditation, something like ISO 9001 2008 in India. So in order to get the accreditation, we need to test all these things. So based on this, again, if the, like say for example, here we need not wanted the entire specimen, we just needed a small portion of like leg or any other tissue, and we can just extract DNA, because that is the beauty of this real-time technique, because very low concentration of DNA can be detected by using this technique. Only thing is, like, we cannot go for a downstream application like for sequencing, but in some time it is possible, because generally we tend to design primers for almost 100 or less than 100, okay, or maximum of 150 we can go. So it's always good to have primers less than 100 base pair product size. Okay, so yeah, so that's how we do nowadays. Like, this is the way of identification system which is in place at MPI PHEL. So apart from that, what we can apart from this modern technique, we currently use some other tools as well. Say for example, something we call in general as isothermal nucleic acid amplification technology, which is something similar to real-time PCR, but only thing is that here we use different polymerases. Okay, and apart from that, here the design of primers are a bit difficult, because here 6 different primers are required. So as I had showed in this image, and all of these things, it'll grow loop in loop, and it'll become like it can give different colors based on its pH. So that's how the determination or the species identification is possible. But apart from that, there are some other technique. Apart from, like within iNets, there are various methods which is available. The first one is loop mediated isothermal amplification, which is shortly known as LAMP, and another one is recombinase polymerase amplification, which is generally known as RPA. Okay, so both these things are working almost similar way. So yeah, these all are advanced technique which we use in our lab for identification purpose, apart from the barcoding which has been extensively used. So that was all on molecular identification and how we can identify species. So the second part is on pest management. So how can we manage this pest group as a whole? Okay, so in that itself, there are several methods which is available, but today I'm going to talk only on RNA interference or which is in short, it is known as RNAi, or gene silencing, we can call in other terms. Okay, so so of course there will be a question like, what is RNAi then? Okay, so which is a naturally occurring nucleotide defense mechanism, which is well elaborated in basal genomes. Say, for example, in case of Caenorhabditis elegans, okay, so where they observed this RNA phenomenon for the first time, and for the same they won Nobel Prize. Okay, so what it means? So as I told you, like these are well elaborate in basal genomes, and then what is it doing in basal genome? So they are controlling the gene expression. Okay, so that's what the answer. And for what they are, why do they need this gene expression control by using this RNAi? Yes, they need to protect their genome, because against retroelements, transposons, and RNA viruses. Say, for example, classical example is bacteriophage attacking a bacteria. There, the protection mechanism, they are using the same RNAi. Okay, so yes, now the process, how it can be processed. So it can be processed by just dsRNA directed sequence specific degradation of mRNA. So now we need to know like what are the different steps, how it is acting in basal genomes and other genomes. So we need to know how can we use this for pest management or any other management aspects in entomology. So coming to the overview of RNA mechanism, as I told you in my previous slide, that there is something called dsRNA. So dsRNA is nothing but double-stranded RNA. Okay, so RNA can be of various types, as you know, like you might are familiar with RNA structures, DNA structures. So DNA and RNA can be like either as like single-stranded or double-stranded. And similarly, RNA can also be like single-stranded and double-stranded. But here for this RNA mechanism, we need double-stranded RNA. Okay, there are plenty of double-stranded RNA which is available in the environment in the form of like transposons or whatever. There will be enormous sequences which like dsRNA material which is available in the environment as well as in the system. Okay, especially when it's like when a virus is attacking, if it has a double-stranded RNA, it used to get injected into the body. Okay, that's how they works. That's how the mechanism works. So as on when the double-stranded RNA which comes externally or internally, so our cells has some enzymes known as dices. So what dices will do, anything and everything which is present in its vicinity, it gets chopped into smaller molecules which is known as siRNA, or in short it is siRNA. Other words it's known as small interfering RNA. So once all this process is happening inside the cell. Okay, so once the siRNA has been formed, there are some ribonucleoprotein complexes that comes and occupy this siRNA molecule, which is known as RISC. Okay, in short, RISC, in expanded form, RNAi-induced silencing complex. Okay, so the RISC protein components will come and occupy the siRNA molecule, which has been produced from the dsRNA by dicer enzyme. So once it has been occupied, which is known as an inactivated RISC molecule, because it has both the strand. All right, so I told you we are going to degrade mRNA. Okay, so how can we get rid of one strand? Because we need to retain only one strand which is complementary to our mRNA. So the RISC complex will get some signals from some of the protein molecule, okay, based on its thermodynamic energy, which we are going to see in the next slide. So they know which need to be retained within the complex, and the other one will just go out of this complex and it'll get destroyed, okay, inside the cytoplasm, where the activated RISC will take it to where its complementarity in the mRNA, and once it's bound to that, the activated RISC molecule along with its core components, it will be able to degrade or cleave the mRNA molecule into pieces. So once the mRNA has been cleaved, then there won't be any kind of a protein. That's how the gene expression has suppressed in a way. Okay, so that is the whole mechanism what is happening in case of RNAi. So now we need to know like how can we use it in insect pest management. So before going into that, so we need to know like the general insect physiology for a little bit. Otherwise, it might be really difficult to understand what and how it is working. So say, for example, it can be divided, the insect body can be divided generally into three parts, head, thorax, and abdomen. So anything which has been taken through the mouth, it generally ends up in midgut, okay, where it has its own epithelial cells along with like the border epithelial cells in the midgut, and whereas it has its own Malpighian tubules, okay. So say, for example, if I'm taking the epithelial lining of this midgut, that is what I showed here, okay. Say, for example, we are going to administer some dsRNA molecule, okay, externally. This is for pest management. So we decided we are going to use a gene. So I'll come to that in detail, but assume that we generally design the dsRNA molecule and which we are going to administer into a cell, okay. So we can administer. So when we are administering, there is something called SID1. So it is known as systemic interference defective 1, okay. So this is the cells, or in an endocytosis manner, this dsRNA can be taken into the cell. So once it has been inside the cell, I told you the dicer is present, RISC is also available, and apart from that, it has SID2, which is a core component for the conduction of silencing signals from cell to the neighboring cell. Okay, so sometime it might be present, sometime it might not be present, but still it can transmit, it can transport the silencing signal by various other mechanism, various other unknown mechanism, which is not known. And apart from that, there is something also known as RDRP. So this is nothing but RNA dependent RNA polymerases. Okay, so what it does is, like I told you, the dsRNA is coming in, so it will be getting chopped up. Okay, saRNA will be formed, and in that I told you one passenger strand will be get rid of the RISC complex. So if RDRP is there, what the cell can do is, by using that template, it can synthesize its own dsRNA inside the cell. Okay, so in turn, what happens is the effectiveness of RNAi gets increased if the RDRP is there inside the cell or in that particular organism. Generally, we'll discuss like how the distribution is like in various group of animals which is existing on Earth, and even in plants, how they are getting distributed. Generally, it won't be there in animals. Okay, I think that you might be like familiar with polymerases. There are different polymerases. In prokaryotes, they are different. Eukaryotes, they are different and different. And yeah, so all sorts of things, like whichever the mechanism, like polymerase, it can synthesize from 5' to 3'. So it's another important thing which you have to retain in your mind. So this is the general things, general physiological points which you have to keep it in your mind. So now we are coming in more in depth of molecular biology of RNA. So I told you that when the dsRNA comes in, the dicer chop into saRNA molecule. The moment it produces saRNA molecule, immediately one of the end, it will be less stable, and other end will be more stable compared to the less stable end. Okay, so that is how they are recognized by various other molecules. Okay, because again, it's all ATP dependent process. Okay, so once saRNA molecule has produced, okay, so as I told, more stable end, immediately the core complex of RISC loading complex, that's what I put it as here, RLC. So RLC is having two components. One is dicer 2 and R2D2, okay, which is again a protein, nucleoprotein. So the nucleoprotein component R2D2 comes and bind to the more stable end, pushing the dicer 2 to the less stable end. Okay, so that is what the first mechanism which is happening after the saRNA formation. So after that, the RISC loading complex immediately gets signal that there needs another complex which is known as holo-RISC complex, where the argonate protein and other enzymes are involved. So immediately, once it's been attached, the holo-RISC complex can form an interaction through argonate protein to the dicer 2 molecule. So once it has been established, the argonate protein sends signal and an unknown enzyme, which is termed as unwindase, which helps in the process of separation of the double-stranded saRNA molecule into single-stranded. And based on the thermodynamic strategy, again, the passenger strand goes out of the RISC loading complex, Okay, only the gate strand, which has the complementarity in the mRNA, which will be retained. And once it is unwinded, like it's being pushed into the unwinded passenger strand will go into the cytoplasm where it will get destroyed, and the retained strand slowly get transferred into the argonate protein of that matured RISC complex. So the mature RISC complex can take the whole of its material, like whole of these protein complexes, to the mRNA, where the single-stranded mature strand can go and bind to the mRNA very efficiently, and then it can cleave, as I told you. Because so I'll explain here how it can cleave the mRNA very efficiently. So as I told, like again, how the RISC functions. So it has several core components, out of which the first one is the PAS domain. So the PAS domain helps the recognition like of the 3' strand, okay, 3' end of the single-stranded siRNA molecule, which has been retained in the complex. Okay, the passenger strand will go away. Only the gate strand will retain. The gate strand, within the gate strand, in RISC complex, the PAS domain holds the 3' end of the single-stranded siRNA. Whereas the PV molecule, the PV complex in that nucleoprotein complex, which can act as an RNase H-like domain. So that means RNase H can chop nucleotides into pieces. So that helps in the cutting of mRNA or just cleavage of mRNA. Okay, and again, PV has another activity which can perform as it can also act as helicases. Okay, RNA helicases, and all of these processes which is happening inside this small machinery, which is all driven by ATP. Again, as I told you, it can the PV domain can cut the mRNA into pieces, but it won't go and generally cut in wherever it as and when it feels. But what it does is it will count the exact 10th and 11th base pair. Okay, so and that too from the 5' end of the small RNA or the retained gate strand. So you can count here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So in between this molecule only the breakage will come. Okay, so there only it can cut the mRNA. So that's how the entire process is happening. So again, as I told, like in case of plants, insect, and nematodes, there are different machinery which are working. So based on that, all of this process varies. Like as in case of plants, it forms a siRNA molecule. It's just like the argonate protein along with the gate strand. It goes and binds the mRNA broken down into small pieces. But at the same time, as I told, the passenger strand, okay, which goes and bind to this RDRP, and it can form its secondary siRNA molecule, which can again go back to the loop and which can do the silencing more efficiently in case of plants because they have RDRP in their system. But whereas in case of insects, which doesn't have RDRP in its system, so what it has, it has normal machinery, so it will form the same way which as we explained now. Okay, along with the argonate protein, the gate strand can go and bind to the mRNA get integrated into disintegrated into pieces, so there won't be any kind of an expression of that particular protein. Similarly, here in case of nematodes, they have this RDRP. So similar to that of plants, they are able to produce the secondary siRNA molecule very effectively. So again, RNA efficiency is much more in case of nematodes compared to insects. So this is a phylogenetic graph. It shows phylogenetic tree, which shows how the distribution of RDRP and SID has been taken place as on when it's organism has evolved. So you can see some of the pretty old insects. Say for example, ticks and C. elegans, which possesses both RDRP and SID. So that means RNA will be much more effective in ticks and C. elegans. Okay, Caenorhabditis elegans. But whereas in case of mammals, in elephant, they have SID, but they don't have RDRP. But in case of plants, they don't have SID, but they have RDRP. So all this information has been gathered from a paper which has been published by Gordon and Waterhouse in 2007. So again, I told you that we need to design some of the dsRNA. All right, so we are going to use it for pest management or insect pest management. So how can we target them? We cannot go and target as on when we feel whatever we as a random pick. Okay, so for that we need to classify the genes into based on its function. So we need to know like which all are the classes which is involved in detoxification functions in insects. So say for example, if in case of detoxification, if you are talking GST, glutathione transferase, CYP450, carboxylesterase, these are the clusters of genes which are mainly involved in the detoxification functions in insects. But whereas in case of digestion, serine protease, chymotrypsin, sucrase, 1, 2, 3 are some of the key players in digestion in case of insects. Again, another classical, like one of the most important processes inside insect development are like metamorphosis, as most of you know. So for this purpose, we need an enzyme which is generally known as juvenile hormone. Hormone known as juvenile hormone. And for the conversion of juvenile hormone precursors into the juvenile hormones, there are a number of enzymes are required. So one of such enzyme is juvenile hormone acid methyl transferase, which helps in the formation of juvenile hormone from its big precursor. Okay, so we are going to see that in the next slide. So again, so if metamorphosis is harmed, then insects can't survive. Okay, so again, we can target something like cytoskeleton, nerve impulse conduction, midgut pH, pheromones, olfaction, all such kinds of things we can hamper, and we can like damage, or we can retard the growth, or we can do like we can target many of its functions, normal functions. If we are disrupting the normal functions of like, then they cannot survive anymore. Okay, so ultimately they will die. So that's what our aim, right? So yeah, so again, then we need to know like what are the advantages if we are proceeding with this RNA strategy. So you might have, like as I told you before, this dsRNA is rather stable than single stranded antisense RNA. So of course, this antisense RNA technology is of like it has gone out of scope as on when this RNAi technology came into the picture. Okay, high degree of specificity is possible because they are like most of this molecule works on the sequence specificity. Highly molecular potent, like they are much more potent and effective compared to like it can be compared with any of it can be compatible with any other insect pest management approaches. Say for example, in case of BT, Bacillus thuringiensis. Okay, and in some cases we can observe like systemic silencing. So that means the spread from cell to cells, and then it can give a very good silencing effect in the organism. And high level safety to the non-target sequence organism because it's highly sequence specific. Okay, and sometimes it's possible that silencing effects could be passed through generations as well. Okay, so and it's also possible that it can be introduced at any different life stages. Okay, it not necessarily that it has to be injected or introduced into the body at very early stages. It's nothing like that. So it can be introduced at any stage of their life. So then now we need to know like, okay, say for example, we have the dsRNA with us, and then how we can supply to the insects. So there are whereas feeding is available, semi-synthetic diet is possible, and even micro-injection is possible for a limit. Because micro-injection might not be a possible way to conduct our experiments in the field trials. But of course, in the lab level, initial trials in the lab can be much more efficiently can be done by using a micro-injection procedure. So again, there might be number of factors which influence the success of RNA in insects. So again, a paper published in 2010 suggests that the concentration of dsRNA is very important. Length of dsRNA is also important. Persistence of the silencing effect, which is another important thing. Like if sometime as on when the dsRNA is available in the cell, which might have some effect, and as on when it gets decreased, then the silencing effect has been nullified. Okay, and then nucleotide sequences, of course, and the life stage of target insect also plays a crucial role. Say, for example, if we are targeting some genes in particular life stage only, it might have its significance. Okay, so we need to know some aspect about insect physiology as well before selecting the genes for RNAi. Okay. So I'm going to explain two two such experiments which we conducted in our study. So the first one was on we did on, again, juvenile hormones, which I mentioned, like in case of metamorphosis. So generally, as most of you know, like insects do have a brain, and where they have a section called corpora allata. So the in the corpora allata, the juvenile hormones getting produced, okay, from its precursor molecule, which has been known as farnesoic acid, which has in turn produced from a series of chemical reaction, okay. And the farnesoic acid get converted into juvenile hormones with an enzyme called juvenile hormone acid methyltransferase, and that gene has been encoded in a length of 1.2 kb size, okay. Say, for example, if we are blocking this particular gene, the conversion of farnesoic acid to juvenile hormones has been hampered, then there won't be any production of juvenile hormone. As a result, complete cessation of larval feeding is happening, no more metamorphosis, insect will die, okay. So say, for example, if due to some reasons we cannot do that, we are unable to do that, then once the juvenile hormone is produced, then it has been transported by two other important receptors or carrier molecule. So the first one is juvenile hormone binding protein, which is of 0.87 kb in size, and where they helps the transportation of juvenile hormone from corpora allata to the hemocoel, okay, where again, like say, for example, if you are blocking this, juvenile hormone is not going to get transported into the hemocoel at all. Again, cessation of larval feeding is happening, death is the end result, okay. Similarly, another receptor which is pretty much important for the transportation purpose, so which is known as methaprin receptor, which is around 1.55 kb gene. So if we are designing a double-stranded RNA for this particular gene, again, cessation of larval feeding and that will be the end result, okay. So in our experiments, what we did was, like, we designed several primers which were required to amplify the gene by using the conventional PCR, and then we need dsRNA has to be synthesized. For that, we need primers to synthesize from its DNA template, which has been produced from our PCR, and it requires T7 RNA, T7 promoter region in its primer. So then only this readily available T7 RNA polymerase can synthesize the dsRNA molecule, okay. And of course, we need to know, like, whether the introduced dsRNA molecule, does it has any effect, okay. It's not only by looking in its morphology or like into its physiological aspect, but also we need to validate it by using molecular studies, okay. So we need real-time PCR primers in order to validate how much it has been suppressed or how much it has been expressed, okay. So yeah, so we designed all of such primers and probes whichever is required for various experiments, and we conducted the experiments. But before conducting the experiments, it's very important to assess that whatever we amplified by using the PCR, is it of correct origin. So say, for example, here in case of JHBP, we proved that in turn it is cladding with another ephid, which is known as acetosiphon paisom, because most of our studies has been conducted on Aphis gossypi for most of these genes, okay. So again, and you can see they are phylogenetically closer compared to several other organisms, which is something like Coleoptera, Tribolium castaneum, Bombyx mori, Lepidoptera, and all those things. So we know that, okay, phylogeny suggests that it is JHBP, because we know that the acetosiphon paisom sequence, which is exactly JHBP, juvenile hormone binding protein. So we concluded that yes, it is something like homologue of acetosiphon paisom JHBP, and we termed it as JHBP of Aphis gossypi. So similarly, we did the same thing for VATPSH, which is another gene which we used in our study, where this VATPSH is very important in the active transepithelial cation movement in case of insects. So if this ion movement, when it stops, what it happens is the pH in midgut gets disrupted, and then it can severely affect the organism, okay. So yes, so we did experiments. So what we did was we amplified the product, whichever of interest. So here in case of JHBP, we amplified almost 500 product size. And from that, by using that as a template, we synthesized the dsRNA molecule by using in vitro transcription kit, which is readily available. And then after synthesis of dsRNA, we extracted it from the gel. Then we purified the dsRNA by using various steps, which has been uh, uh, which has been proposed by various companies. And then what we did is, after the dsRNA production, what we did was we conducted a bioassays using aphids. So here, again, we need to know bit of insect physiology. So say, for example, in case of aphids won't come and drink. Okay, so what it does is, it has a very strong stylet. So what it does is, it penetrates through the xylem and it will reach the phloem. So that means they are phloem feeders. So we need to know, we need to devise such kind of an experiments that the stylet can be penetrated in, and then from there it can take up whatever the material, the diet. So what we did was, we synthesized the dsRNA molecule and mixed it with sucrose diet. So again, from cotton research, they were knowing this diet formulation, and then they given that, and then we made the sucrose diet formulation. Along with that, we incorporated the dsRNA of various concentration for various treatment methods. And then what we did was, we made a sandwich of parafilm feeding method. So we put the constituents, including sucrose diet along with the dsRNA in between the parafilm. And then what it happens is, this aphids can go and probe into the first parafilm, and it will take the sucrose diet very efficiently. Okay, so here you can see, as I showed you here, you can see very clearly the control aphids were looking very healthy, but whereas the treated one, you can see most of them are dead inside the treatment. So here again, you can see, this is with VATPSH, you can see the body, especially the body part has been shrunken quite a lot compared to the control insects. So again, not only the physiological changes, we haven't studied on only the physiological changes, but also what we did was, we calculated why as the silencing is happening inside the body of this treated aphids, and we could find that the percentage silencing was directly proportional to the percentage mortality. So as on when the relative expression comes lower, it has become more significant percentage in case of mortality, and mortality went up to 60% after 96 hours. And similarly, the downregulation of the gene has almost reached 30 to 40%. Okay. So similarly, here again, percentage silencing of JHBP, juvenile hormone binding protein, has showed that significantly the expression has reduced, and similarly, it has reached extensive death rate, percentage mortality up to 60% after 96 hours with a concentration of 50 microgram per microliter. Sorry, 50 microgram. Okay, it's not per microliter. So again, our study has showed some clear indication of the presence of systemic silencing in Apis gossypi. So that's how we concluded was. Say, for example, as on when the shows this graph, if you are looking, you can see the percentage silencing of VATPSH, which was almost constant throughout different treatment and different hours. So here in 48 hours, you can see almost the maximum was almost nearing 60, and 72 hours, again, it was on par with 60. And again, come to 96, it was on par of 60, 65. Okay, but whereas in case of JHBP, you can see as the concentration increases, in 48 hours, it reached maximum of 25%. And again, as the time progresses, it went up to 35%. And as when time progresses to 96, it went up to 75%. So this proposed, because of these attributes, we propose that, okay, there could be a possible mechanism which is operating in aphids for systemic silencing, the existence of systemic silencing for the first time. Okay, and yeah, that's how it works in case of genes which are regulating in various ways. Okay, but apart from that, apart from the mortality, we thought that, okay, we need to demonstrate that it is not only works for, the pest management cannot be only devised for such kind of mortality causing genes, but can be used in some other factor as well. So what we done was, we selected some other genes generally, which is a key player in case of olfaction. Okay, so which is generally known as odorant binding protein. Okay, so what this odorant binding protein does is, it helps in the insect olfaction and determining various chemical cues from the host plant. Okay, so then what we did was like we again amplified the gene from Apis gossypi and we made its own chemical structure by using FIRE software. And again, as I told, like as I explained in the previous slides, like again we went up to like the phylogenetic analysis where we proved that okay, it is within the homolog sequences of like OBP2, and we understood that it's a classical OBP2 from Apis gossypi, whatever we have isolated. And again, what we did was we completed our real-time studies here because we won't get any kind of a mortality as on when we silence this OBP2. Okay, somehow the insect will survive. Okay, but we need to know like how whether it has been affected the organism and its activity. So what we did was like we silenced the OBP2, and then what we done was we went and studied the electroantinograph response. Okay, so what it means, so we will remove the head portion of the insect, and by using the like removed head portion, we will see its antennal response against various chemical cues. So what we used was, one was sucrose, which is a diet. Second was honey, which is again a natural feeding substance. And third one was the cotton plant where it feeds on. Okay, so it will get some, like it will produce some chemical cues in that, and it will respond according to that. And again, the fourth one was indole, okay, which is very important in oviposition procedure for insects. Okay, so and we calculated the percentage silencing. So we know that it was known from the previous experiment that 24 hours was so much not effective. So we went up to two different time intervals, 48 hours and 96 hours. And from there, what we understood yes, the relative expression was significantly lowered as on when the time progressed, and it went up to like almost 80% down regulation. Okay, and correspondingly, you can see the EAG response by these insects has also showed clear down response for various chemical cues. And as on when the time progresses, as on when the higher concentration has happened, like was in place, you can see it showed very significantly low EAG responses. Okay, so here in this like EAG response studies, we have only used the 96 hours treatment this thing, where compared to the 48 hours, it was showing significantly lowered down regulation. Okay, that's why we used 96 hours. And then we went and studied the EAG response. It was showing again the relationship was almost directly proportional. And this is the graph which shows how it has responded. So you can see the lac is at control, which was showing a clear peak that it, as on when we given the cotton plant, it was showing a spike. And like if the video was available, I could have showed that. I like as on when I searched, I couldn't find a video. So you could have seen that the antenna was moving quite quickly. Okay, so that shows that the dsRNA lac is at control, which showed significant responses. Okay, and but whereas the treatment, like it was not showing good responses, you can see compared to the response, like compared to the control, it was showing very poor response. And similarly, in case of sucrose, honey, and indole, it was all showing, the treated ones were showing very much lowered antenogram responses. Okay, so in a nutshell, what we can conclude is like the diet-mediated knockdown of target gene is an invaluable tool in insect pest management. Okay, so apart from that, the AGOBP, Apis gossypi odorant binding protein 2, which that study has showed that it plays this particular gene plays a crucial role in host seeking, detection of oviposition attractants. Okay, because we use indole, so we made the conclusion that it is significantly playing a role in oviposition attractants. And yeah, as a result, what we can propose is like not only the mortality causing genes can create significant impact, but apart from that, something similar like OBP kind of genes can also potentially serve as a practicable target for RNA-mediated gene silencing in hemipteran insect. Okay, so again, since all of this work has been done on aphids, that's why it's been proposed on hemipteran insect pest control. So it doesn't mean that it works only on hemipteran, but of course it can it can be used in many other pests as well. So yeah, as a senior scientist here in New Zealand, so I'm currently working on various projects. So the one which is very important study was on life after death, where we are trying to track the exact time point of death by using various RNA molecule which is available in insects. So that is one of the interesting project which is going on. And apart from that, we are also using iNETs technology in conjunction with eDNA, environmental DNA, as a modern biosecurity diagnostic toolkit to detect insects as quickly as possible. Okay. And apart from that, as you know, like GMO is becoming like more and more, it has been widely used. And yeah, we are heading a project where we are finding various ways to detect GMOs which has been imported into New Zealand, because New Zealand is not promoting GMO till date. Okay, so yeah, that's another project which is undergoing. So that's all. Thank you.
Thank you, Rebijit.
Right, thank you.
That was a highly informative and resourceful presentation.
Yep.
So now this session is open for discussion. So I have requested the participants to post their queries in their comment box.
Yeah.
So I'm waiting for the queries from the participants. Okay, the now session is open for discussion. Participants, dear participants, the session is open for discussion. You can directly ask your queries. Anju?
Yes, sir.
Do we have any queries in the YouTube platform?
No, sir, not yet.
Okay. So participants, if you have any doubts or questions, you can ask right now. Anju, look at the chat box.
Yes, sir.
Please read out that question.
Okay, first of all, thank you, Rebijit, sir, for this informative session. And a question here, and it is from Mabel Varghese. The question is, why nuclear DNA studies less accepted than mitochondrial DNA? Is it because of the lack of introns in the mitochondrial DNA?
No, it's not that way, because the thing is that, see, as I told, like CO1, because it has been proposed that it has more of a, like what we can call like evolutionary more important, and also it can throw more information on its diversity aspect. Okay, so that is one thing. And again, yeah, of course, as she rightly pointed out, it has quite a lot of introns, and it's been very tricky to work with nuclear gene compared to the mitochondrial genes. And most of this, like say for example, in case of animals, it's been proposed that CO1 is the most accepted, and it has quite a lot of sequences which is already available in NCBI and BOLD database. So people use that one as the a particular molecular marker or DNA barcoding marker. But it doesn't mean that we can't use any other nuclear marker. There are several other nuclear markers which is currently available and which is used in molecular identification of certain particular species of insects. Okay, so but generally what happens is, like, as long as we have a very robust method which is available, so we generally tend not to go for a nuclear marker. That's what the answer. But it doesn't mean that, in say for example, in case of plants, they use extensively like both nuclear marker as well as mitochondrial marker. And sometimes some of the plant species or some genera which might not be useful at all if you are using mitochondrial genes for DNA barcoding. So at that time, they have to go for some of the nuclear markers.
Okay, sir, we have another question from Anusruti Ramakrishnan.
Yeah.
The question is, sir, can you briefly explain about the phylogenetic tree preparation? Which are the softwares used and do we always need an expert to do it?
Oh, it's a very, very interesting question. But the thing is that I might require another session to explain on that aspect. But in generally, like say for example, if we have sequences of like CO1 or any other genes, so what we tend to do is, in simple words, we can say for example, when we are working on barcoding, we can make an NJ tree by using mega 10. The current version is 10, so we can use mega 10 and you can create an NJ tree which might be more sufficient to get it published. But when we are looking from a phylogenetic aspect, it might be really tedious, and then you might require an expert there, because we need to know like different aspects of phylogeny. So there, what we require is like we need different softwares. Why we need is like we need to clearly understand how the evolutionary procedure has been happened within that species, whatever we have studied. And as I pointed in my talk, like it might not be a correct idea if you are using with one particular gene. Rather, we have to use a combination of markers. If possible, the best way is combination of mitochondrial as well as nuclear markers. Say for example, something like CO1, CO2, or NADH from mitochondria, and elongation factor and ITS 1 or 2 from nuclear gene. Okay, and then we have to do a test which is known as then we need to do an model test, which is generally to conclude that all these data, can it be combined or not. Sometimes it might be possible to combine. Why means, like as I told you, like it might have different evolutionary pace at which it moves forward. So sometimes the data set can be combined, sometimes it might not. So in that case, a simple aspect, we can do a model test. Either you can do it in either POP or J model test. So there are various softwares which is available, and then we need to assess the phylogenetic evolutionary relationship by using various methods which is currently available. Something like maximum parsimony by using POP or some other software, and apart from that, ML values, maximum likelihood by using RAXML software. And apart from that, apart from all of these things, the most important and the most trusted one is the Bayesian method, so which can be run by using Mr. Bayes. And apart from that, if you need to look into various aspects like how it has been diverged, okay, so that such kind of dating studies can be used by another software which runs again in Bayesian platform, which is known as BEAST. So a combination of softwares and techniques need to be put in place when we are doing a good phylogenetic studies. So where we need like a better understanding on phylogenetic analysis as well as softwares, and also the terms which we use in phylogenetic research papers. So that's all. And in my case, like generally what, as I told, like different softwares has been employed. Like this for barcoding studies, I opted the simplest and easiest one is the Mega 10. From there, I created the NJ tree. Okay, that was the simplest one. And apart from that, as I mentioned, in phylogenetics, I used a number of softwares like J model test, POP, then maximum likelihood in RAXML, and then Mr. Bayes for Bayesian tree, and BEAST for deciphering this dating of divergence. That's all.
Okay, sir. Jitin Johnson is asking, how good is it use the H3 histone coding gene complementary to the commonly used nuclear gene and a mitochondrial gene? How good is to use the H3 histone coding gene complementary to commonly used nuclear gene and a mitochondrial gene?
Yeah, you can use histone marker as well. Like H3 markers has been employed for identification in certain group. The question is like, I cannot suggest like how far it is worthy, but then in order to understand that how far it is worthy, we need to do the experiment and we need to validate. But generally, histone gene, as I know, like it's a small gene sequence. I think it might be 300 or 400 in case of insects, so which might not be a suitable thing to decipher molecular identification or phylogeny.
Okay, so next question is from Priyanka Prabhakaran.
Yeah.
What is the interspecific and intraspecific distance range?
Oh, interspecific and intraspecific range. So intra means within. Okay, so say for example, like as I mentioned in my slide, like if I told you like there are three different. Say for example, let us take in the example of Thrips palmi. So I told there were two groups, that is Indian population and the rest of the world population. Okay, so Indian population. So within that Indian population, how far is the genetic divergence? So that shows intra. Okay, within. And similarly, we'll calculate Indian population, how much is the intra-specific divergence, and similarly we'll calculate it for world population. So we will be having two values, say for example 1 and 1.1. Okay, and then we will calculate the interspecific distance. That means the Indian population versus the world population. We'll calculate that genetic distance, and if it is something 11 and above, then we can call they are cryptic in nature. Okay, because Y means the intra-specific is 1.1 is the maximum for the world population, but whereas the interspecific distance is almost 11, that is 10 times more, right? So in that case, we can call that it's a cryptic species. So that is what, as she asked, like intra means within this particular species, and inter means between the species.
Okay, sir. Gayatri AM is asking, what about miRNA? Can we use it instead of miRNA for slicing?
Oh, again, miRNA is another great topic. Like again, it depends. Like, see, as I explained here, even in my PhD, I did quite a lot of work on miRNA as well. So yeah, you can use like RNAi is a general technique. Okay, you can use, say for example, you can use double-stranded RNA mediated gene silencing, and then siRNA molecule can be used as for inducing silencing, and also miRNA can be used. Okay, so but all of these small RNAs, which has its biogenesis pathway. So the enzymes which are involved in these biogenesis pathway is slightly different, as in case of as I explained in case of RNAi, there the dicers are coming into the picture. But as on when we move back into the miRNA, again the enzymes are changing, which is known as DROSHA and PASHA, which are the key enzymes in the biogenesis of smaller like miRNAs in general. Okay, but again there are various ways of formation of this miRNA, which is not yet fully understood. Around 90% it might have been fully understood, but again still there are black boxes yet to fulfill their complete biogenesis pathways. Okay, so then again there are various types of miRNAs, even like within small RNA. Within miRNA there are various kinds. Again, if it is from the classically known miRNAs are the classically known ones are again 22 to 25 nucleotide. Again, if it is slightly higher in size, like something 30 and above, then or 27 and above, which is generally known as pi RNAs, which is known as PV RNA. So yeah, any of these molecules which can use for inducing silencing. So as I told here, like in our experiment, we used double-stranded RNAi mechanism, but you can use, of course, miRNA can also use for inducing silencing and further down regulation. Okay, so yeah, that's what the answer.
Okay, sir. We have a question from Dr. Binu Ramachandran, and he's asking, how do you perform the antenographic recordings?
Oh, so that's again EAG. So which when I was doing my experiment, we had another institute called National Bureau for Agriculturally Important Insects, NBAIR previously. Now I think it is some other name. Okay, anyway, it's a bureau for insects which is situated in Bangalore. So they had a specific instrument called EAG instrument. So what we do is like we will cut the head portion of the head portion of the insect along with its antennae, and we'll fix it on into an electrode, specific electrode. Okay, so we'll like small small vibes of electricity when it passes, and we will at the same time we will just shoot our these chemical stimulants. Okay, so then we'll come to know that electroantennogram, which is producing any kind of signal as on when its antenna is moving, so that will record against this what is that, chemical cues. Okay, so yeah, that's how we used to do that. And yeah, there is a specific instrument known as electroantennogram. Okay, yeah, I hope I answered the question.
Question is from
Hello?
Nida.
Yeah.
She's asking
Am I
how
audible to?
Yeah, I think Binu is asking some query. Yeah.
Hello, Dr. Davis. Thank you very much for this highly informative talk. And for this antenographic recording, where do you place actually the electrode in the?
Where do you place the electrode? No, the electrode. We will fix the head portion into that electrode.
Okay, no, I mean basically this is an automatic recording from the which portion actually? This is a field recording, yeah, from the head portion, yeah.
Yeah, it won't. Yeah, see, again, you have to do a lot of series of experiment in order to stabilize it, because yeah, as you rightly pointed, it might it might shake its head, so that head movement also can be recorded. But if you are properly fixing its head onto the electrode, then you will come to know that you might not be taking the entire head as a whole. Just you will cut it immediately, just immediately fix it onto the electrode, and yeah, then you will come to know, like, as on when you're fixing, the head might slightly change here and there, but yeah.
Next question is from Anushruti Ramakrishnan. The question is, can you suggest a best primer to do DNA barcoding studies for family Culicidae?
Oh, it's highly difficult to propose that way. Yeah, because yeah, as I told you, like, barcoding means, yeah, you can, of course, first marker which coming into my mind is COA1, cytochrome oxidase 1. Try that. If I think 99% it will work. If your luck is that bad, only you might require another primer. But in that case, even you have plenty of markers within mitochondria, something like COA1, COA2, and then you have something called 28S marker from nuclear gene even. Okay, so yeah, somebody has asked, like, why can't we use nuclear genes? Of course, we can use nuclear genes. In 28S, we have several fragments known as D2, D3, D4, D5. There are several regions. Okay, so we can use many markers. But for DNA barcoding work, generally in case of insects, 99.99% COA1 will resolve the problem. Okay, otherwise you can try several other markers, as I pointed out, COA2. Then 28 is D2, D5 regions, D2, D3 regions, and so many other markers which is available. And in the slide, I pointed out something like different markers which is available for insects, plants, and bacteria and fungi.
Okay, sir. We have a similar question from Shiva Supriya. Which is the best primer to do DNA barcoding of Diptera?
Again, Diptera we all use COVAN. Okay, so yeah, we 99.99% species can be identified using COVAN. There won't be any kind of a problem.
I think we have no more questions left, sir.
Yeah. Okay. Right. Thank you.
Okay, so Abhijit, you have answered every question in a convincing and reasonable way.
Okay.
Thank you. So we are coming to a close of our webinar. So now I request Ashwati, the research scholar of Shatpada Entomology Research Lab, to deliver the vote of thanks.
Good afternoon. I, on behalf of Shatpada Entomology Research Lab, deem it a great honor to propose a vote of thanks to all who have helped us in making this webinar a success. First of all, I would like to propose hearty vote of thanks to Dr. Abhijit for his enlightening talk on perspective of molecular approaches in entomology. The way he explained the topic is exemplary. Thank you, sir, once again. I also wish to express my gratitude to our principal, Reverend Dr. Jolie Andrews, Manager, Reverend Father Jacob Nirinampilli, Vice Principal, Dr. K.Y. Shaju, Vice Principal, Reverend Father Joy P.T., Dean of Science, Dr. Tessie Polpi, Vice Principal and HOD of Zoology, Dr. C.O. Joshi, Dr. Robin Ponminisheri, IQAC Coordinator, Bursar, Father Vincent, and all the faculty members of Zoology Department, Dr. Sudhikumar A.V., Dr. Leon Wadis, Dr. Abhishek, Dr. Sister Dilla Jose for their encouragement and support. I express my thanks to Dr. Bijoy C., Assistant Professor and Research Supervisor of Shatpada Entomology Research Lab, for his continuous efforts to make this webinar happen. Now, let me thank all the research scholars of the department, especially Ajit V. Johnson, Surya Narayanan TB, and Anju Sara Prakash for their indomitable spirit that made this event reality. I would like to thank the technical team for their effective and efficient backup, making this virtual platform happen without any interruptions. I extend a word of gratitude to all participants for their cooperation and patience. Once again, my heartfelt thanks to all of you. Have a great day. Stay safe. Stay healthy. Thank you.
So thank you so much once again.
Abhijit sir, thank you so much for being with us for the past 3 hours, spending a lot of time for the Department of Zoology, Christ College. Thank you.
Thank you.
Okay, sir. Thank you so much.
Thank you. Bye. Yeah, bye.